Folks, if I could have your attention, welcome to the September 4 edition of Charlotte Rotary. And I hope you all had a wonderful long weekend. And to get us underway today, we've got Mac McCarley with our invocation.
I invite you to join me in a moment of thanks. Lord, we thank you for all the blessings in our lives, and as we come to this hour, we especially thank you for these friends, this fellowship, and this food. Amen.
Thank you, Mac. David Soroy is going to introduce our guests and visiting Rotarians. David.
So the first visitor is Carol Jagu, guest of Stephanie Heinrich. Jeff Dockett, guest of Kim Bratton. Mike Torty, guest of Hank Donahue. Lauren Bremer, guest of Wes Jones. Bill Crowder, guest of Benton Bragg. Joe B. Martin, guest of Rhonda York, nephew of Governor Martin. Sandra Moore, guest of Luther Moore. Khalif Rhodes, guest of Brian Karn. And then Sean Prophet, guest of Kim McMillan. Okay.
Great. If everybody else would join in standing, we'll do the Pledge of Allegiance. I pledge allegiance to the flag
of the United States of America, and to the republic which stands, one nation under God, indivisible, with liberty and justice for all.
Thank you. You can be seated. David Soroy is one of our technology people. I don't know if you noticed, but he had a paper in one hand and his iPhone in the other, and I think the paper was pretty much superfluous. Next is Matt Joyner, who's brought to us by the composite committee, and is going to talk to us briefly about history.
Thanks, President Mike. When David Zimmerman asked me to be on the composite committee, I thought he was asking me to tell a historical joke in song, but no, separate these things out. So today it's just history. And just look briefly, I'm going to date myself now and refer to Sidney Harris, if y'all remember the Chicago columnist who used to occasionally write things I learned while looking up other things, and y'all, for those were his best columns. Anyway, this is something I learned while looking up something else. Well, I was doing some research on the relationship between Charlotte Rotary and the Mecklenburg County Boy Scout Council. Came upon these minutes from the 1920 meeting in August of 1920, and was just amazed that the Committee on Boys' Work was not only a committee, but it had no fewer than 14 subcommittees, each with a chair and presumably other members. And one of the first one was YMCA, the second one was Boy Scouts. So it's just crazy. So think about all the committee work that goes on, but all of that devoted and all that specification, just crazy. But the person who was behind it is somebody, is sort of the hidden part. I said, well, who was behind all this? A guy named Rogers Davis. How many people recognize the name Rogers Davis? Luther does. Y'all need to know this famous Rotarian, Charlotte Rotarian. He was a charter member of our club. He was the first president of the club. He was elected president of the club in December of 16. That first group of officers served to July. They got reelected for a whole nother year. So he served for a year and a half as president, okay? He then went on to be district governor. First district governor this club has ever produced. And back then it was district number 7. We're now district 7680. So you can see how Rotary has grown since then. And back then, District 7 comprised South Carolina, North Carolina, and Virginia. So he'd been in charge of Rotary clubs in 3 states, okay? So the crazy thing is, this Boys' Work Committee that's being promoted in August of 1920 was his retirement gig after being president for a year and a half, district governor for a year, and then taking on the Boys' Work Committee that had no fewer than 14 subcommittees. This guy was just a major Rotarian and somebody we all need to know about, be aware of, and be thankful, because that's what this club was built on, efforts by people like that, contributing just an incredible amount of time. So next time you feel put upon, serving upon a committee, think about Rogers Davis and all that he did for Rotary. So thanks.
Thank you, Matt. Actually, duty on the composite committee is not all that bad if you want to volunteer for it. Jesse Height's going to introduce our newest Rotarian.
Thank you, President Mike. It is my pleasure today to introduce to Charlotte Rotary our newest member, Howard Layton. Now, he is not new to Rotary. He is new to us, but he is a member, has been a member of the Cabarrus County Club and has recently moved to Charlotte, and he will be joining us. Originally from Detroit, Howard is a graduate of both Wayne State University and the University of Michigan. No, I'm not gonna say that.
Let's be nice.
Yeah, I'm not gonna say that.
I saw the game too.
Okay.
Let's speed ahead, gents.
Yeah, we were gonna keep going. Anyway, he obtained his MBA from the University of Michigan and then went on with a 30-year career with Ford Motor Company, where he was involved in many senior management levels and such things as positions in vehicle planning and scheduling, material procurement, re-engineering and process improvement. He was also involved in Ford's international operations, and he was responsible for the strategic planning and development of the marketing aspects of the Ford and Mazda partnership. He also served as the Chief Information Officer with Ford Credit Company. After leaving Ford, he went on to serve as a Director of Worldwide Automotive Consulting with Coopers & Lybrand and Deloitte Consulting, where he advised clients throughout the world on strategic planning, business development, and supply chain management in international organizations. In addition to his automobile career, he has been an educator where he's developed programs and taught both undergraduate and master's levels programs both in the U.S. and internationally. In 1999, Howard founded his own firm of Layson Logistics, and he provides consulting services in the independent and executive advisor role. His primarily focus would be on strategic planning, project management, market and financial analysis, where he can use that knowledge that he gained from those many years in business for his clients. He was very active in the Cabarrus County Club, and he looks forward to being involved in Charlotte Rotary. So let's welcome our newest member, Howard Layton.
Just a quick reminder that this is the last week to drop off school supplies, either in kind or if you want to leave a check, loose change, cash, whatever. Karen Calder, I know, is here today, and you can leave that with her or with Sandy, and we'll get it to the right place. So, introducing the head table of prominent Rotarians, to my right, Bill Costangi, and you heard from Mac McCarley. Bob Freeman, of course, over at the far left, and to introduce our special guest and speaker today, Carla Dupuy.
Uh-oh. I'll leave it there. Take a moment to think about what honor means to you. Our speaker today was raised in the church, the son of a Presbyterian minister, so I'm guessing the concept of honor was ingrained in him at an early age. To those of you familiar with Davidson College, you know the honor code is a way of life that creates the atmosphere for trust and love and, yes, honor. As an elected official, our speaker was the Honorable Commissioner, Congressman, and Governor. Join me in welcoming our friend who lives with honor and serves with honor, the very Honorable Jim Martin.
Well, thank you, Carl. I have to say it's an honor to be here. After I had retired, I'm going to leave out my jokes, by the way, because I have a couple of things I want to get across to you before we leave here today in mass. After I'd retired, I wanted to do something, and I started writing a book. I'd been giving lectures on the subject of science and religion, and I had these wonderful slides that I'd copied off of the internet. You can do that as long as you're not commercializing, but you couldn't use those in a book because you never know who really owns the copyright. If someone claims it, you send them a couple $100 for a right to use their illustration, and then you publish the book, and then you get sued for a couple $1,000 by the person who actually has the copyright. So you'll see at the bottom of the book It's going to be a little hard to read. Anyway, we recruited our son, Jim Jr., to do about 70 out of 77 illustrations in the book. You'll see some of them here today. Some of them are humorous. That he has the structure of an atom, the nucleus at the center and the electrons zipping around outside like a Pac-Man cartoon. So that's easy for him. But that made it different. I'll come back to why the lightning strike there with the pink side. Now, my purpose is not to tell you what you should believe, but I think it's fair that I share with you what I believe about the subject that I'm going to discuss. I believe that the Bible is the revealed Word of God in narrative and poetic form. I believe it is the best guide to faith and practice, but it is not a science textbook. I believe that modern science gives us the best approach, the best opportunity to understand how God did it. And that's basically what this subject is about. Now, you don't have to believe anything that I say, but you will get, that is except for the facts, you don't have to have the same belief that I do. You can leave here with the same belief you came in with, but I'm going to give you a lot to think about. Now, that's a little scary because that means you got to stay awake, right, as we go through this. And it means I've got to move through some things that ought to take 10 minutes on. I'll get maybe a minute and a half, but that's what we'll have to do. The title of the book, Revelation Through Science, as you see, is based on my belief that science reveals to us. It's a modern form of revelation. It doesn't tell us anything about who God is, but it tells how God created the universe and created created life as much as we can understand about that. Now, for centuries, there was no problem. Most of the studies of nature and naturalism were in the universities that were run by theologians. But then along comes Galileo, 1609. He read about this little toy spyglass that was being sold in Holland, and because he knew a lot about optics, he started making his own spyglass and looking at the sky. He was the first person to do that and report what he saw, and it was astonishing. Blemishes on the moon. But in this one, the overhead lights may be killing the effect, but on the right there's Jupiter, and to the right and left of it are a couple of moons that he saw, that as Jupiter moved across the panoply of stars from night to night, these four little lights moved with it, but moved around it as it went, just like our moon does. And he thought this was more consistent with Copernicus' idea that the sun was at the center of what we could see. It's not really, but at least the earth goes around the sun rather than the sun going around the earth. Though we still talk about sunrise and sunset, don't we? But on the left is a photocopy of his notes that he took over about 12 nights showing the different positions. But his biggest discovery was about Venus. Sometimes you see Venus in the morning when it comes up before the sunrise. That's the morning star of poetry and opera. And sometimes you see it as last night or tonight, setting some 30, 40 minutes after the sun, but it usually is somewhere in the vicinity of the sun. Now that was a difficult problem for the Aristotelian scholars because they believed that each of the moving objects, the sun, the moon, the 5 planets that they knew in that day, were moving around the earth in crystalline spheres that held them in their track. And the problem for Venus was that it seemed to be moving around a little point somewhere between the earth and the sun. Well, Galileo looked at that idea and he said, well, if that's true, and I look at it with my little telescope, spyglass, it should always be a crescent. It'll never be half moon or never more than half moon. But when he looked, he actually saw sometimes it was crescent and sometimes it was half moon. He couldn't see it when it's full or dark because that would be in line with the sun. He'd go blind. You don't want to look at the sun through a telescope. It's not a good idea. But you can look at Mars with your binoculars if you have some good binoculars. They're twice as powerful as his telescope. And you may be able to see those out in the field of your binoculars. But this was astonishing because it meant that if you believed in those crystalline spheres, Venus was crashing through the sphere of the sun. And he got into a lot of trouble with this as he tried to explain it. In 1617, he was hauled before the Inquisition. They told him, you can talk about your theories, but don't say they're true. Okay, you can talk about them and you can use them to show how they might explain what's going on, but don't say it's true. But his good friend, Pope Urban VIII, wanted to encourage him. He said, go ahead and write a book about this. And he did. And he said, while you're at it, because Urban fancied himself to be an amateur astronomer, and he believed in the older Ptolemaic idea that the earth was at the center and everything was going around the earth. He said, while you're at it, you're welcome to use some of my writings on the subject. That was a mistake, because he had these two characters, one Salviati and the other Simplicio. And he put the Pope's words in the mouth of the simpleton Simplicio. And that got him into real trouble, hauled him back before the Inquisition, 1633, where he was sentenced to house arrest for vehement suspicion of heresy. Think about that. Vehement suspicion of heresy. Well, his remains after he died were kept in a box, sort of nondescript, in one of the off chambers of a church in Florence for about 70 years, and then the Doge of Florence was able to persuade the church, let us unpack that box and put him in a proper resting place. And what you see on the left is the current sarcophagus in which Galileo, his historian, and his daughter are all buried in there. And it is easily the rival for magnificence of the one that you see on the right of Michelangelo, which is in the same church. Just thought you might enjoy that. Well, things settled down for 150 years as people began to adjust to the earth going around the sun, and was about to get into a new round of dispute when young Charles Darwin, with the assistance of his family and friends, was able to get an appointment as a naturalist on board the HMS Beagle, which was assigned to go around the world and measure the depths, chart the entrances to harbors all around the world. And this was a great opportunity for him. When they get down to the Falklands at the base at the south end of South America, he was told about these little cluster of islands that each one had a different kind of fox. And the natives of the Falklands could look at a fox and tell you which island it came from. Well, that was very interesting to him. Why would there be a different kind of a species of fox? Well, they're all the same species, but a different variation of fox on that island. Then they get up to the Galapagos, and you see that's where the giant tortoise, he saw each island had a different kind of giant tortoise. And the people there could look at the animal and tell you which island it came from, because they were endemic to a single island, each little shape of, and there were also birds, finches, that had different beaks depending on what kind of food was available. And he came up with this idea of natural selection. He believed that there was something going on that caused a change, a variation in a species. He didn't know what it was. He had no real way to reflect on that. But he said once there's a change, then nature selects which one is most suited for the environment, for the amount of food, for the availability of others of the same species, for predators, the ability to escape predators, whatever it was, would enhance the chances of that surviving. And so he developed his theory of natural selection, which we now know is evolution. And this one is just for vertebrates, just showing his second book about descent of man was really about descent of all species from common ancestry. And this was based on not only what he observed, but on reports that others had made of fossil finds. He had actually found some fossils on his trip, but he used those of other people and came up with this fascinating idea, which today is the centerpiece. It is the basis of modern biology. And for a long time, as long as we were talking about fossils and comparative anatomy, gross anatomy, it was easy for atheists to say, for example, and some of you may have this view, to say, if you believe in evolution, you don't need God. And so there's no need to believe in God. And so some Christians, in order to fight back, fundamentalists, said pretty much the same thing. Well, if you believe in God, you can't believe in evolution, which assigns evolution to be the property of atheism, which it should not be. They were saying you can't believe in both God and evolution. I will say to you, I believe that you can. And I'm going to show you a couple of things. You're out walking in the woods, and you see a turtle sitting on a fence post. You know it didn't get there by itself. Okay? That's the end of my, in conclusion. Astronomers had been able to measure distances to remote galaxies. That was Edwin Hubble. And another of his colleagues measured the shift in light toward the red end of the spectrum, which meant they were moving away from us. All of the galaxies moving away from us. And what Hubble found when he compared his findings and Slipher's was to see that the distance was proportional to the velocity at which they were moving away. If one galaxy B is twice as far as galaxy A, it's also moving away twice as fast, which is why it's twice as far away. If you take that back in time, 13.7 billion years, all matter in the universe had to start, he concluded, from the same point. Well, to think it's a point, I'd say it's bigger than a breadbasket, but that's a debate for another day. But this has interesting aspects to it. What came before that Big Bang? It was called Big Bang, and someone was trying to ridicule it, but now that's generally accepted by astronomers. What came before that moment, 13.7 billion years ago? Science, it has no way to ask the question or to answer it, so what you have there is a theological question, regardless of what your belief might be. Physicists then began to point out that there were a number of physical constants, about half a dozen of them, one of them being gravity, that were so finely tuned that if any one of them was slightly stronger or weaker, we wouldn't be here. For example, gravity, if we're slightly stronger, think of this, by 1 part in 100 billion, 1 part in 100 billion, the universe would have collapsed shortly after it burst. The gravity would pull it back in. If the gravity were slightly weaker, nothing would have condensed into stars. We would all be hydrogen gas escaping out into the sky. There would be no stars, no planets, no galaxies, just hydrogen. Be a nice fuel, but nobody to take advantage of it. Now, it was also known even in Darwin's day, but a lot more known about it today. Just look at the red line on there. That seems to fog out a little bit for me. But this is the Cambrian explosion that happened a little over 500 million years ago. The Earth is 4.5 billion years old. But before 500 million years ago, there were only 4 phyla, P-H-Y-L-A. That's a body form. A phylum is one body form of animals and plants. There were only 4. Bacteria, single cell creatures. Jellyfish, if you've been down to the beach recently, you might have had some acquaintance with that. Sponges and roundworms, that was it. And then all of a sudden, in the span of 10 or 15 million years, a blink of an eye in geologic terms, there suddenly appeared, without much in between, some 6 or 7 dozen new body forms. And there's been a lot of effort to try to explain how that could happen all of a sudden. Said, well, maybe all of a sudden there was oxygen, and so you get all these new animals. Well, there was oxygen before that. There's been oxygen since. And any attempt to explain how that happened has a fundamental problem that it has not happened one time since. Not one new phylum has appeared in nature since 500 million years ago. And some that were around then have become extinct because they didn't produce enough species to carry on their line. So we're down to about maybe 4 dozen. One of the paleontologists, fossil hunters, who has found many soft shell fossils in China, Chen Junwan, was visiting the United States talking about some of his finds, and he said, you know, it's funny, in the United States, you can criticize your government, but you can't criticize Darwin. He said, in China, we can criticize Darwin, but we can't criticize the government. 1953, the year I graduated from high school and started college, I later learned about it. There was a famous experiment by Harold Urey and his student Miller. Urey already had the Nobel Prize for having discovered heavy hydrogen, deuterium. It's hydrogen with not just a proton, but a proton and a neutron at the center. It's not going to be a test on this, so don't worry. I'm just trying to show you there's something special. And he had gotten the Nobel Prize for that, but his student Miller wanted to look at something he'd read that had been predicted by a Russian physicist, and they took this globe and had in it a mixture of gases that they believed would be present in the early Earth atmosphere. Carbon dioxide, water vapor, nitrogen, and methane. That's carbon with 4 hydrogens around it. Methane, carbon dioxide, water, and nitrogen. And they ran an electric arc through it, just like a lightning strike. And rather soon, within a few minutes, it started to turn pinkish brown. And when they analyzed it, they found a collection of amino acids. They hadn't put any amino acids in there. They were formed by the high energy breakup of those few simple molecules that they believed would have been in the abiotic, life-free air. Amino acids, why is that important? Because they are the building blocks of proteins. And we've got maybe 23,000 different kinds of proteins in our bodies. Each one of them, as we'll see, has its particular role. We won't go into all of them here today. But one reason I wanted to show you this is because that's why the cover of the book commemorates the Urey-Miller experiment. But if you have amino acids, does that give you proteins? No, because proteins have the amino acids aligned in a very precise order. They're not randomly scattered. They're in a very precise, measurable order. And for a long time, biochemists struggled to try to unravel the sequence of amino acids. There are about 20 of them that are incorporated into protein of all plants and animals. That same year, James Watson and Francis Crick reported in a little paper about 3 pages long in Nature magazine how they had figured out the structure of DNA. Now, by then, it was known that DNA had something to do with heritable characteristics, with genetic characteristics. They didn't know how. But they're standing there with a model of it that was famously described as a double helix, going 2 helices wrapping around each other, spiraling from the floor upwards. And they had this scale model that was, well, let me show you one more picture. 40 years later, somebody got them to get together for another picture. This time he didn't have a slide rule, so he used a ruler. But he got a little more sophisticated model there. You can see it spiraling. I hope you can see that spiraling. Yeah. There it is. Very simplified drawing. The ribbon-like part is just phosphate and a sugar, deoxyribose. That's why it's called deoxyribonucleic acid. And in the middle are the nucleotide bases that bridge across. And the reason they were able to figure this out is that they had a paper that had not been published. One of their colleagues gave it to them, and it said there are these 4 nucleotide bases, adenine, cytosine, guanine, thymine. And he had done quantitative analysis, and he said that thymine, for example, has a different abundance in the DNA of different animals and plants. But in every one, it always has exactly the same abundance as adenine. A and T are always exactly the same. Cytosine and guanine would be somewhat different, but always from the others, but always identical to each other in their quantity that's available. And they figured that that meant that there was some association between adenine and thymine and between cytosine and guanine that could not be mixed from cytosine to thymine, it had to be A and T or C and G. And the way in which those could alternate going up the rungs of a ladder gives you an enormous number of possibilities that constitute the code of DNA. Human DNA, tell you how many rungs it has. It has 2.1 billion rungs. And one of the questions then is, how could it assemble that code very precisely? Francis Collins is the head of the National Institutes of Health, was previously head of the Human Genome Project, has written a book called The Language of God. It's a metaphor, language of God, the code of DNA, the language of God. Interesting, because he started out as an atheist and self-converted reading C.S. Lewis' Mere Christianity, which somebody suggested to him. So here, this was a simplified model of it, and those drawings on the right are representing at the top adenine and thymine, the bottom guanine and cytosine, reading from right to left. You don't need to memorize these, but you see that the ones on the right have two rings, a 6-membered ring, a 5-membered ring. The ones on the left have just a single ring. All of them have nitrogen atoms. All of them have oxygen. And there are these weak bridges across that fit the exact space between those two helices going up. Now, this was significant because it helped explain replication of DNA just before cells divide. And what our cartoonist shows, you can see what's left of the DNA down to the left side of the picture. But on the right, you see what happened as that talon zipper comes back through the DNA. It unravels it, and it goes off with the different strands no longer bonded to each other because of the effect of this enzyme called helicase, or it's a cluster of enzymes called helicase that unzip. So the talon zipper helps you to understand what's going on better than you would if I kept it entirely technical. But while it's doing that, another little bunch of enzymes called polymerase start picking up pieces and rebuilding a duplicate with each of those strands, a duplicate double helix around each one of those strands, because it can only attach adenine where there's a thymine. It can only attach G where there's a C. Remarkable theory to be able to do this. It also explains transcription of where RNA, that's ribonucleic acid, has ribose in a single spiral, single helix. Ribonucleic acid gets its code from one gene of DNA. So your genes in DNA give their code to RNA, ribonucleic acid. That's transcription. And now this one's a little complicated, but the main thing, look, that's a protein factory called a ribosome. See the little smokestacks? To help you understand, that's a factory. And the little train going across the bottom is the messenger RNA with the code from one gene. And that code prescribes the sequence of amino acids one at a time that are going to go into the protein that's built by, I said you have 23,000 proteins. That's because you got 23,000 genes that have the code, each of them, for just one protein. And each one of them gives its code to an RNA, which then is functioning to get the amino acids into the little protein factory. And so today, while Darwin didn't know how the changes took place, now because of the understanding of what DNA is and its code and what it can do, when there's an error, that's when you get a variation, when there's an error that can't be repaired. All of this, then, DNA studies have validated, as far as scientific proof, Darwin's idea of evolution. Now, they're still working on it, as Chen Jun Yuan like to say. We're still trying to work and prove it, to learn more about it and how it can work. But what I would say here at this point is that while on the one hand it provides in scientific terms absolute proof for evolution, at the same time it reveals to us, it has revelation of something so inordinately complex that you have to wonder if life could ever arise by unguided chance. You may conclude whatever you want to conclude about that. That's up to you. I've got time for one little side trip here. One of the things that Francis Collins found in his study was that humans have a defect in the genes that control your biting muscles. And it's the only species that has survived with that defect. It means our biting muscles are not as strong as those of all other animals. And he's able to determine that this happened about 2 million years ago. And when you take the skulls of fossils from back 10 million years, you find that an interesting thing began to happen 2 million years ago. The brain cavity began to expand. Was that perhaps related to the fact that we no longer had those strong muscles to crush the brain down and keep it in small size? Right up to Homo sapiens at about 1.5 liters. Will it keep going? Hang around, we'll see, or somebody will see. I'll give you an illustration. If you have a dog or a pet and you want give it a treat, put your hand on top of the dog's head and you'll feel these powerful muscles on top of the head, just piling, no matter what it is you give them. Soft piece of meat, whatever. Those powerful muscles are crushing. Now bite down with your hand on your own head, you don't feel a thing. Above here is about as far as you go when you chew. Come on, you can do it. Everybody can see. Bite down, you can feel it here, but not up here. That Francis Collins is a brilliant man. So was this a lucky 1 in 100 million coincidence, or would this be an intervention? Theological question. Well, now, like Lady Godiva, we're getting close to the close. And on this one, this is the most difficult thing to comprehend. But it's made easier if I tell you, look at your hands. And from time to time, you can look at mine. Roll them like this, and you'll see they are mirror images almost, warts excluded. Okay, fingernail broken. But they are mirror images, but they're non-superimposable. If you've got a first baseman's mitt and you're right-handed, that only goes on your left hand. It won't go on your right hand, right? Did I get that right? Non-superimposable mirror images. Now, amino acids, that's what's represented there. You can see something up there, COOH, that's the acid, carboxylic acid. You see NH2 in one of the little parts, that's amino. H on the outside is hydrogen, and then the R varies from one amino acid to another. If you have 100 different things, that's 100 different amino acids, only 20 of which get built into proteins. But they are also non-superimposable mirror images. Now, why is that significant? And by the way, if you can't see that in the drawings of the model, look at the little hand there sticking out from the R. The one on the left side is clearly a left hand, right? The one on the right side is clearly a right hand. The one at the bottom is the one on the right side turned around on its vertical axis 180 degrees. So what you see when it turns around, it doesn't become a left hand, it becomes the back side of a right hand. Okay? Now, why is that important? We can synthesize all of these amino acids in the laboratory, and when we do, we get equal amounts of the left-handed and right-handed amino acids. In nature, every protein is assembled exclusively from right-handed amino acids. Excuse me, got that wrong. Left-handed amino acids. Disregard that previous. Every known protein is assembled exclusively, see, I was testing you, exclusively from left-handed amino acids. Why? Right-handed amino acids are available. Well, maybe it has something to do with RNA. The colored figure in the middle is a computer-generated image of DNA spiraling. Do you see it spiraling in a right-handed fashion, like right-handed wood screws, righty-tighty, nuts and bolts. On the left, there's a left-handed thread above at the top, right-handed threads at the bottom. You go to your hardware store and you ask for some left-handed wood screws, they think you're crazy. They don't have any. They wouldn't dare have left-handed nuts in the racks with the right-handed bolts because people would get furious because they wouldn't go on. Chrysler once made automobiles with left-handed lug nuts on the left side. They thought that was clever until people started complaining they was stripping the threads trying to get the tire off so they changed tires. Somebody at the beginning of the Industrial Revolution decided we're only going to have what we call right-handed nuts and bolts and screws. Could it be that somebody decided early in the industrious evolution of life that we're only going to have right-handed DNA and right-handed RNA? And if RNA is right-handed, maybe it only picks up left-handed amino acids. If it was left-handed, it would only pick up right-handed amino acids. So we get left-handed amino acids in the protein because RNA is right-handed. Why is it right-handed? It's because the sugar, ribose, is right-handed. Sugars are complex. They are mirror images, non-superimposable mirror images. We can synthesize them. We get equal amounts of both. But in nature, there are no left-handed sugars. They're all defined as we do as right-handed. And those right-handed sugars on the bottom right, ribose and deoxyribose are right-handed. They cause the RNA and the DNA, the single helix and the double helix, to be right-handed. That causes only left-handed amino acids to be picked up to form proteins, and the sugars, ribose and deoxyribose and every other sugar, are biosynthesized in the cell by what? By protein enzymes that are left-handed. You see, Newt Gingrich likes to talk about these triangular relationships. And we talked about, how here's the voters and here's the Democrats and here's the Republicans and how the relationship, well, these three are interdependent on each other, like a three-way chicken and egg. Which came first? My conclusion is they all had to be there right-handed or left-handed as appropriate, with all that enormous code in the RNA, in the DNA and RNA, in the first living cell, or it would never have reproduced. Something to think about. If you're interested, books will be available outside. I guarantee it was tough, and I'll tell you, don't try to read more than one chapter at a time. Don't try to do all at one. But when you do, you get to a point and you say, well, this is amazing. It seems miraculous, but I don't fully understand it. Just say, wow. Wow, and go on. Okay? Good night. Bye.
Thank you, Governor. Bob Freeman asked me before lunch if I would report to him as to how much of this I understood, and a whole lot more than I did when I took organic chemistry, which brought me to my knees. I knew I should have gone to Davidson to take this course. But anyway, we got time for some quick questions. Yeah.
Or challenge.
Challenge, yeah.
Governor Fred Brown, you remember I've caddied for you a couple times. We were actually playing golf.
He didn't give me enough strokes. Is that your comment?
I was out in the woods so much. Believe it or not, the governor's, in addition to being a great leader and an academician, a really good golfer too. And another small thing, small world, I'm going to get to my question.
Now's the time, Fred.
And Mike, I ran hospitals all my life. The first hospital was a little town of Siler City, North Carolina, and I hired Francis Collins to come and do
Wow.
moonlighting in our emergency room.
That's the NIH director we were talking about.
And he's, I had one of my doctors said, he's so smart I can't understand him. I read, I mean, if you believe this, and I do, that, our vision is so finite and it's hard to believe the infinite. I tell my little grandsons, and I can't remember who wrote this, that if there's, if
that right?
this world is habitable, and it is, at least by some of us, there's likelihood in the universe of having other places that are habitable by life is greater than the grains of sand that exist in this world. I'd be curious if
In
we
other-
don't have to.
That I do know, the Fermi paradox. Can you comment on the Fermi paradox?
Yeah, Fermi said if there's so many planets out there with life on them, where's everybody? And of course, we've been searching over the last dozen more years for planets. They found some 6,000 planets, and they only find the ones that are in a orbit that we're in the plane of their orbit, because we see them as they go across their star. The star dims about 3%, and then it brightens again. You wait a couple hundred days and see what happens again. And so they found them. But there are only about a dozen that are just the right size and just the right distance from their star. It's like Goldilocks. If you're too hot or too cold, you're not going to have life. If you're too large or too small, you're not going to have life. If you're too large, you're covered with water. There may be life in there, but there's not going to be technology that we can communicate with. So where's everybody? We may find them, we may not. It will be the single most dramatic and amazing and significant discovery in,
All humankind.
Yeah.
Yeah.
well, humans go back 200,000, so 200,000 years.
Yep. Edwin Peacock going to close us out.
Governor, can you explain the chemical makeup of the leaders of North Charlotte? It doesn't seem like the left hand or the right hand is talking to each other as it relates to toll roads. You drive on this toll road issue. Talk to us about the chemical makeup up there.
Well, I would say part of the problem is those on the right and the left, but part of it is all of you in the middle who abandoned your party, and now there's about the same number of Democrats and Republicans and unaffiliated, which means the parties are even further apart. They don't have to talk to each other. So those of you who pulled out of the party, you choose your party, you get back in your party, try to moderate your party, do what you can. I mean, when I first registered, I registered Republican as a protest because you were told you can't be a Republican because you don't get to vote in the primaries. And so I protested. My parents were Democrats, my wife and her parents were Democrats. I was a protest. I just figured we needed a two-party system in the South. Well, maybe we've gone too far. But then the center pulled out and made it worse. And we like our little silos. That's not chemistry, but that's what you're talking about.
Does that do it for you, Edwin? Very good. Let's give the governor a big round of applause. Thank you, Governor. And the governor would be glad to talk with you about anything, politics, chemistry, whatever. He's going to be out in the back.
Selling books.
Selling books and signing books, even better. So next week, our meeting falls on September 11. We're going to have a panel discussion, which is going to be a bunch of prominent folks, including Governor Pat McCrory, who was the mayor of Charlotte at the time, Sheriff Irwin Carmichael, Medic Joe Penner, Chief of Police Kerr Putney, there we go, and Fire Chief Reginald Johnson. Ed Driggs is going to moderate it. We are likely going to be in the smaller room, slightly smaller room down at the end of the hall. So if you really need to be here for this, but come early and come with others if you can, because the parking's going to be a little tight. But we will see you next week, and we are going to start at about 12:40, so we're going to start a little early without announcements and go right to the program because we have so many prominent folks on the panel and so many things to say. So with that, Governor Martin in the back, hope you have a great balance of the week. We're adjourned.